we're all a bit Zoom weary, I'm sure, but I'm convinced that we can have valuable and deep scientific exchanges over video conferencing, and of course it saves on jet lag and carbon footprint. I'm really happy to be here and delighted to take questions at the end. I have very fond memories of visiting you all about 10 years ago at the time, and so it's interesting to think about how one's ideas have changed in that time. I did open that presentation back then just to see what I was talking about, and there are some similar themes. And one key theme is somewhat an obsession with trying to do biodiversity science that makes a difference, and that makes a difference in the context of the huge footprint that human beings are having on our planet. And when we think of the idea of the Anthropocene, this denomination of the period in which humans now dominate the Earth system, we often think of images like this, right? We see agricultural transformation, land cover change, the sky and the lights in the sky at night, and we think of merging antimicrobial resistance or eutrophication or habitat loss, and all of these images that are so evocative of the Anthropocene, one can notice that they are happening at different scales. We're observing them happening at different spatial scales and at different temporal scales, and the mode by which these human changes on our planet are happening are unfolding at different scales all the time. And so one of the core idea of my presentation today is that we need to tackle this head on, that we can't just be thinking about biodiversity science or its application at one particular scale. And to do that, we need a mature approach to thinking about how to observe responses at different scales, but also how to understand the intrinsic scales of ecological systems as they're responding to these pressures. So for example, we can think about ecological change that comes about when we apply a pesticide, or and the immediate ecological impacts that might have, and how that application would cascade through a food chain. But we can also think about the longer term evolutionary responses to selection, right, as exemplified here. But we know that antimicrobial resistance is just one iconic example. We now know that evolution and ecology are happening on a sort of contemporary timescales. And so I think a mature science requires that we think about how these pressures are operating. An integrative biodiversity science requires that we think about how these pressures are operating together at these scales. But ecologists think about scale often in the notion of space, right? How spatial scale is observed and how we detect patterns as we go from the micro to the macro. And so we also have to build on the progress that our field has made over the last 30 years in thinking about the fundamental nature of scale. So here's a quote from Gordon Orions: "One of the great challenges in ecology over the next decade will be to build conceptual bridges between micro and macroecology." And that was in 1980. So when I think about scale, and when we think about scale, we often have this kind of notion that Google now provides us so easily, maybe as we're flying across a continent. We think about human impacts at the scale of the eastern seaboard of the US and Canada, and we can see agricultural transformation and land use change. But then as we zoom into an entire ecoregion like the St. Lawrence lowlands here, where you're seeing agricultural transformation and forest loss, we also see a city and a conurbation of 3 to 4 million people and urban sprawl extending out past its into the countryside. And as we zoom in, we see patches of remnant ecosystems, human land use transformation, and high urban densities. But ecology is still important in this context, right? And now you're seeing the urban canopy in the town where I'm living, and that little red house there you see in the middle is where I'm coming to you today. I'm sat in this little red house, and I'm struck by the fact that we've lost 70% of ash trees in our street. So we're seeing the manifestation of an invasive pest that now has continental or semi-continental extent, and the impacts it has at a very small scale that affect me as an individual. So a mature biodiversity science really needs to be able to tackle the various dimensions of this across the scales that we've just flown through. And the other side of it, of course, is not just about the basic fundamental biodiversity science, but it's also about the application of that science to conservation and policy. And so what we know is that so far, if we think about these axes of time and space, we know that the policy arena has been primarily either at very small spatial scales and temporal scales, or at very large global scales. When we think about the Convention on Biological Diversity, for example, or major actors in like the IUCN or the major NGOs, they're often taking a global perspective, which is built on these ideas of linking the health of ecosystems, the functioning of ecosystems, to the changing status of biological diversity. But when we think about the evidence we have that links biodiversity change, for example, to ecosystem functioning, it's almost all of it at the scale of a few tens of meters to a few hectares. All right. And when we think about the impacts of anthropogenic drivers on biodiversity, the conversation is often at that global scale, or certainly at regional to global scales. So what we're pointing out is that we need to fill this data gap. We need to fill this translation of science at the local and the global scales with this missing middle, this sort of intermediate scale from local to regional, as we stitch together the evidence that we need to make informed decisions at the scale of landscapes and seascapes that are often natural units for policy and governance. So what I'm trying to do and working with collaborators is to fill this missing middle and think about how can we, as Gordon Orions pointed out, link the micro to the macro. So my talk today really is about doing that, showing you some of the progress we've made from a sort of theoretical standpoint with some experiments, but also in part one, which is where I will focus on the work we're doing right now on expanding the relevance of biodiversity and ecosystem functioning research from small plot scales up to regional and then up to global scales. In the second part of my talk, I will then shift gears to think about the application of connectivity conservation science to decision making. How can we use our best science on ecological connectivity to really inform policy and decision for land planning that not only maintains ecological connectivity, which is so essential for biodiversity, but also for restoring it in areas where it's been eroded or lost. And if I have time today, I just want to update you on some news that Geobon has come to Canada, and I want to do my best to engage the Canadian community in this huge global network that's dedicated to establishing a global biodiversity observation system that we need if we're going to cross these scales that I've been emphasizing now for the last few minutes. So let me start with the first part of my talk. Right, I want to give you a bit of an overview of a paper that we published just last year that tries to synthesize everything we know about the links between biological diversity, its changes in space and time along these two axes here, from the local to the global, or from the scales of minutes to centuries. We know that biological diversity modulates the way that energy is used, nutrients are recycled, decomposition happens. We know that happens at very small scales, and most of the data represented by these little black dots here are in that box on that far back left-hand corner of this figure. But what we need to do if we're going to reach out to the global, if we're going to provide relevant information to the global policy arena, we need to know whether the information and the knowledge that we have acquired so far can be expanded up to these more lasting or extended spatial and temporal scales. Now, when we do that, we know that other things change. It's not just a question of the scaling, it's also a question about the biological structure of the assemblages of the ecosystems that we are studying. All right, so we know that often at small scales, we're dealing with transient or small assemblages. In BEF, it's often been about, in biodiversity ecosystem functioning research, I'll call it BEF from now on, is often at a single trophic level and focused on plants. There has been a much more work on food webs in the last decade, but we've often greatly simplified the system. And we've certainly rarely considered what happens when we extend to multiple assemblages interacting as a meta community or as a landscape of systems, or on up to the macro system, right? To entire continental stretches of biomes, and thinking about how biodiversity contributes to the functioning of those at those scales. So the effort in this paper was to summarize what we know from theory and from data, and then take it from there, figure out what are the next rounds of theory, empirical science that we need to expand our scales of inference. And of course, as a terrestrial ecologist, I often ignore my marine colleagues. Sorry, Boris. And I've added here, obviously, that all of this is relevant to the same problem in the marine system, where the scales go from minutes and hours all the way up to global changes in ocean currents and ocean structure and ocean temperatures and the field of oceanography at large. So as I said, BEF has focused on short-term studies to really grapple the empirical basis of BEF. And now 3 decades of research has led to multiple data syntheses. And on average, and I'm greatly simplifying, what we notice is that when you manipulate biodiversity, some functional diversity measure or a species richness measure, whatever the measure is, we tend to notice that there is a decelerating but increasing function between describing the relationship between ecosystem function and biological diversity. When we actually manipulate this as an independent variable, we elicit change in some measures of functioning. Okay, so that's great. That means that we have a piece of knowledge that we did not have a few decades ago, but of course it's much more complicated than that. And this simplified picture of the slope of the BEF relationship being roughly 0.2, of course, that hides that behind it a picture that suggests actually that when we look across hundreds of experiments that have been done now on 5 continents and many different types of ecosystems, we realize that actually there's a distribution of slope values. Of course, there's a central tendency, but there's variation here that you see that can even become negative in a small frequency of cases. This slope can go the other way, is explained by significant biotic and abiotic variation, right? So there's clearly something that we are leaving out or missing out if we do short-term experiments, all right? And we know that some of those factors are quite understandable, quite logical from our point of view. There's factors like the duration of the experiment, the spatial scale of the experiment affect the quantitative estimate, but also trophic layer, trophic structuring matters a lot. And factors like functional diversity, what the nature of the functional groups that are being manipulated, all of these factors conspire to mediate this information that we have, that the distribution of the relationship between biodiversity and function. And all of that at the smallest of spatial scales. And some people have already expanded this application, this knowledge of the relationship at small scales to quantify the role of biodiversity at the largest spatial scales. So here, I was not involved in this study at all. This is Liang et al. In Science, where they took the local estimate that you see in pink there on the top left and said, well, let's model that at the pixel scale for global forest productivity. And then we'll map, based on our modeling, the change in the spatial distribution of this slope. And here you can see in color how the slope estimate, right, the sort of the elasticity of this slope estimate, how it's changing geographically or at this global scale, how that mediates the role of biological diversity, in this case, tree species richness, to the overall quantitative production of the forests. And when you do the back of the envelope estimate, the numbers are huge. So clearly, biodiversity, when seen through this lens is playing an enormous role in the functioning of forest ecosystems. But again, this is an application of the local scale knowledge accumulatively across the planet. When I think about scale and what I want to address today is actually, okay, we let go of a small grain or a fine resolution, and now we start to think about aggregating our understanding. What does that imply for how biodiversity mediates functioning? Is it scale dependent? So let me give you an example. So here is a made up cartoon of a network of ecosystems. Maybe this is a landscape of forest patches or grasslands. Pick your favorite ecosystem, and one can study the relationship between functioning and diversity at different scales of time. One can extend the duration of our study, like Tillman and Peter Reich and colleagues did at Cedar Creek. And what they discovered was that the quantitative relationship between these two variables depends on time, depends on the extent, the temporal extent of the study. And so we don't have a static, stable, single slope of this relationship. It has something to do with the way the environment is changing over time and how this ecosystem is embedding post-sowing of these plant communities, how it's embedding and how it's developing over time. But another view is spatial, not just temporal. So now we can ask, oh, let me stop. I added this little animation here to draw your attention to the fact that the contribution of diversity is changing over time. This is really crucial. So this is the half saturation constant, right? How many species do you need to get 50% of the productivity? And of course, now you're seeing that half saturation constant is increasing over time. Let me now switch to space, and very recent research is showing that now when you extend to entire landscapes, so now I've extended you see here the scope is now for the entire landscape, we're going to look at how functioning happens over space. So now what we have is the number of communities, not the number of species. And the number of communities, how that mediates the diversity required to achieve 50% of ecosystem functioning. And it turns out that what you need to achieve at the scale of an entire landscape, so this is work by Rachel Winfrey's crew at Rutgers, where they've been looking at how many pollinator species, wild pollinator species, you need for effective pollination, in this case, to achieve 50% of pollination across that spatial extent. These are images taken from their science paper. What we noticed that you need, that should be up to there, but you need a large number of communities. Why is that? Well, that's got something to do with the beta diversity. That's got something to do with the turnover of species from place to place, right? We're not compositionally identical pollinator communities. And so the simple fact of having spatial turnover means that now we need to take into account larger spatial scales and how they mediate the strength of the BEF relationship. So the question of scale in BEF is a combination of temporal and spatial considerations that we need to integrate and understand. So in this paper, we identified 6 core hypotheses about what we need to test to establish when and where scale is most important, okay? And so in this, I won't go through all the hypotheses in great detail, but I list them here so that you know that they're out there. And these are the kinds of factors that lead to strong scale dependence. By that, I mean that we cannot just rely on our local scale understanding to make an extrapolation, all right? So in the first case, we know that a nonlinear change in the BEF slope across spatial locations leads to slopes that are varying, like we saw in the Liang et al. Science paper. And when you start averaging over many different pixels, you get another scaling relationship that starts to happen. Another hypothesis was that there's a strong relationship or scale dependence between ecosystem stability and spatial extent, which is linked to the synchrony of species fluctuations over space and time. We know also that coexistence doesn't happen just at the smallest spatial scales, that the factors explaining coexistence often have a strong temporal and spatial component, which mediates biodiversity and the turnover among sites over time, which is critical. And then we know that a key predictor of turnover that I've just shown you was so important for the pollinators and for the plant communities over time is actually to do with how the variance of the environment gets distributed over time and space. That's called autocorrelation, right? How similar conditions are over time and over space. And the scale of autocorrelation, the scale of similarity of the environment will determine the scale at which turnover happens. So if we want to understand turnover in space and time as a crucial ingredient to the scaling of BEF, then we need to understand how patterns of similarity in the environment are changing and how humans are changing that autocorrelation. The fifth is about the connectivity among communities or meta-communities. So when we take a regional approach, we know that the fluxes and the movements of matter, of energy, and of individuals and genes mediate functioning. And that is crucial. I'm going to return to that in the second part of my talk, but it's important to explicitly take into account connectivity if we want to understand how to scale BEF. And then last but not least is that we've tended to ignore the vertical diversity related to food web structure, right? So that could be trophic chain structure, it could be patterns of connectance or modularity. Those factors change in space and time. And so if we want to understand how biodiversity, trophically structured biodiversity mediates functioning, then we need to account for that and start studying it and how trophic structure is mediating the cycling of nutrients and energy, et cetera, et cetera. I'm going to focus very quickly on the intuition around autocorrelation, because this is something I've been working on for quite a long time now, and I think it's crucial to understand for the BEF scaling problem. So let's quickly show you a cartoon about thinking about how we quantify scale dependence. Okay, no real data here. This is just a figment of my imagination. So imagine we take some transect over time or over space. All right, so we've got a linear one dimension here of the environment, and the environment is wiggling, and it could be temperature, could be humidity, precipitation, it could be anything. What we know is that species tend to sort themselves according to environmental conditions. And here you're seeing how species at different trophic levels, the occupancy or the presence of those species shown by a bar here maps to the environmental conditions. All right, we know there's some spatial covariation or codependence among primary producers and consumers. We can quantify the diversity of along this transect using a battery of statistics for diversity and the estimators we need to do that reliably. And then of course we can quantify ecosystem functioning as the relationship to the activity, the functional activities of these species as they map to the environment. And what we're interested in BEF is knowing the spatial covariation or the temporal covariation between these two patterns or fluctuations. Now, key to this question of scale is how we observe it. How fine a grain of observation are we going to take? And so we can take the question at one spatial scale, we can study it at 1 grain, for example, I don't know, 100 kilometers by 100 kilometers or 1 meter by 1 meter. And we'll get one answer for the correlation or the covariation between biodiversity and functioning. But we can aggregate. We could go to another scale of space and time. And as we aggregate, we're integrating over all that covariation. And so now we're going to get another estimate of the interdependence between biodiversity and functioning, between this wiggle and this wiggle. And so it turns out that if you have strong scale dependence in the correlation between biodiversity and functioning, then the observation scale will be crucial. So you need to know both things. You need to know the intrinsic scales of covariation, and you need to be explicit about the spatial scales that you observe the system. So here is an example. I'm now taking a measurement at this first scale, and I'm showing you the correlation, a made-up correlation between them. So every time we have a dot here, we have an observation of the covariation between diversity and function, and we get some slope estimate. right? So at this scale, I'm showing you here the half saturation constant for this relationship. But let's aggregate now. And if we aggregate, we get another estimate, all right? And that is telling us that there is scale dependence in the BEF. And our challenge in the field is to unveil the relationship between scale and the explanatory power of biodiversity as it mediates functioning. So here, what you're seeing now, notice I've put scale, observation scale on the x-axis, and we've got 2 scales. I've measured it at 2 scales, so we've got some sort of linear increase in the importance of biodiversity to functioning. But unless we make a scale explicit approach, we don't know what this will look like beyond this sort of veil of observation scales. So this, in a nutshell, is what the BEF field needs to be doing, is figuring out the explanatory power of BEF as we expand our spatial and temporal scales. And this would be important if we want to disaggregate or we want to downscale back to the local. So this unveiling of the scale dependence of BEF is really where I think we need to be putting some time and effort over the coming decade. So I did promise you that what's crucial here to determining the strength of this scale dependence is how autocorrelated these fluctuations in the environment are, right? This is a very slow varying environment, but you could imagine that this environmental variation is much faster, right? And now species are going to have to track that variability. Well, way back when I did some work on this, back when I was in Paris, at the University of Paris, and we were thinking about how the scale of autocorrelation in the environment mediates function. So imagine here we have, this is a simple model. We were using sort of a classic MacArthur or Tillman resource competition equations. And here we're showing 3 niches, right? We've turned the niches on their side. And here you're seeing 3 species and their preference for temperature that's varying over time here in these gray oscillations. So there's a warm adapted species and a cold adapted species and a sort of an intermediate environmental condition species. And this model basically maps the niche through the environment to give you the biomass over time. And here I'm showing you the summed biomass of the productivity due to how these 3 species are interacting with the stochastic environment, right? And in this case, there's no correlation through time. There's no autocorrelation. It's just white noise. And you'll notice that the species are all roughly equally abundant and they're all contributing to biomass at these very fine temporal scales. But now if I crank up the autocorrelation and I make the conditions correlated over time or space, but here it's over time. So see, now you see there are some trends in the environment. Everything else is constant. We haven't changed anything else between these scenarios, but now you can see the contribution of species is changing quite dramatically, right? We're getting these episodes of outbreaks in which one species is doing well when the conditions fall well within its niche for a long period of time, and then the conditions change and another species picks up. So we're starting to see the signature of autocorrelation on the turnover of the environment, the turnover of the diversity and the composition of these communities. So this is the underpinnings of the scale dependence in BEF. And then last, here is a case where the autocorrelation is extremely high, right? And the conditions of the environment stay similar for a long time on average here. And now we've got this extreme case of these outbreak dynamics that one species come to dominate and stays there for a while, and then the system flips again to another species. What we tend to know is that in ecology, as in geography, any geographer will tell you that the environment is almost universally autocorrelated. And so in general, in ecological environments will tend to be on the right-hand side of this spectrum, which tells us that something about the environment is crucial to understanding turnover and BEF scaling. So we recently just published a paper in Procroy Soc that shows how this autocorrelation mediates the scaling of the BEF, right? And I obviously don't have time to go into the details of this paper, but I do want to show you the 2 key results from this paper that link back to this BEF question. So there are a couple of figures in this paper. that basically make the point that the number of species required to maintain function increases fastest with scale when the environments have low autocorrelation. All right, so here is species richness on the y-axis, and we have spatial scale. And spatial scale is simply the aggregation of patches, like I showed you on the cartoon a few slides ago, right? We can go from one patch all the way up to aggregations of 80 patches, right? We're sort of cookie cuttering the landscape, if you like. And here we're only varying the spatial correlation or spatial autocorrelation. And so you'll see that the contribution of richness, so you see we accumulate species very quickly as we increase scale here in low autocorrelated environments, right? When the environment is wiggling a lot, we see a lot of different species over time and space, well, in this case over space. And when we slow the environment down, we get these slow accumulations of diversity because it's at that larger patchiness that we start to see different species coming to dominate, all right? And this happens I think it's quite substantially different between it. The effect size is quite strong here of autocorrelation. But at the end, the number of species that is maintained in the system is pretty similar. All right, so at the regional scale, the gamma diversity is fixed, but the way that they accumulate over space depends strongly on autocorrelation. But then over time, we get a similar story, right? You see that it's the high autocorrelation environments are accumulating species over time more slowly than when we have no autocorrelation. But there's a slight difference in the temporal gamma diversity, how many species you see over time at the largest temporal scales, because in autocorrelated environments, the environment is much less likely to maintain diversity, right? You saw these large oscillations. So we're showing how autocorrelation is mediating this turnover. And so here is the final result, if you like. What we're showing is that more species are required to sustain the same amount of biomass per unit area or unit time over larger scales compared with small scales. So that autocorrelation is mediating the BEF relationship quite dramatically, right? Here you're seeing the half saturation constant of BEF. So how many species do you need to achieve 50% functioning, in this case, biomass? And you'll see that we see a very strong signature that we accumulate the contribution of diversity to functioning very rapidly at small spatial scales when the environment has low autocorrelation. And when we have high autocorrelation, that very slow variation here, the contribution of diversity to BEF extends over much longer spatial scales. So larger spatial scales are really important for maintaining function per unit area when we have high autocorrelation and large grained, coarse grained patchiness. That is true also in time. The results are qualitatively different because you see that in fact we have trouble maintaining functioning to the in autocorrelated environments relative to non-correlated environments. So even if you don't take away all the details of this, it's very important to realize that the way that the environment is experienced by species at different spatial and temporal scales will determine the compositional turnover and in turn the contribution of diversity to functioning at larger and larger scales. So why does this matter? Well, it turns out that under climate change, we are seeing a strong autocorrelation of the environment. We're seeing a stronger reddening or a stronger slowing down of certain types of variation. There's a recent study a few years ago where they showed using GCMs, they took 21 GCMs and ran time forward from 1871 to 2099 and showed that the climate is the residuals around the mean trend in climate are becoming more autocorrelated, which would suggest from this theory that I've been talking to you about today is that we really need to take into account this trend due to climate on how biodiversity is contributing to function. I'll skip that. So a take home from this part of the talk, that at small scales, relatively low levels of biodiversity can sustain ecosystem functioning. But higher regional diversity is necessary to maintain functioning over larger and longer scales where environmental heterogeneity is greatest and where we're seeing strong signatures of human impact. Okay, so that's the key part of the science that we need to get good at doing if we're really going to show the relevance of BEF research to this global policy arena. Okay, I'm going to switch now. Let's take off our cap of BEF researcher, and I want to show you how we can move from maybe some of this ivory tower research, this sort of fundamental ecological research about what maintains biodiversity, to the application of biodiversity science to land planning and management for ecological connectivity. This is work that I've been doing now for about a decade in Canada. And so what we have come to realize, I think, is that connectivity conservation has come of age, that there is now a genuine openness at provincial, federal, and at municipal levels for land planning for conservation, for the conservation of ecological connectivity. And this is a huge opportunity for our field to get involved and to provide science-based advice. Here you're seeing a map from Nature Conservancy Canada, which is showing you their sort of large arrows of connectivity for this eastern part of Canada here. All right, as we go all the way, down the St. Lawrence lowlands from Toronto all the way out to Charlottetown and the Gaspé region. And then there's a view that is even more ambitious, which is the entire eastern seaboard of the US and Canada, which is wonderful and inspiring, but the science for that needs to catch up. So what I've been working on over the last decade is thinking about how to apply connectivity conservation to design the landscapes of the ecoregion that is called the St. Lawrence lowlands, right? And I've been working with the Quebec government, a couple of ministries of the Quebec government, to think about how to maintain connectivity whilst land use change is happening in this area. And it's obviously a very fertile agricultural zone, but there's also a lot of urban sprawl happening as these big cities like Montreal and Quebec are tending to expand. And cities in this region are also concerned about the connectivity of urban ecosystems and the kinds of science that we can apply to improve ecological outcomes in cities that have obvious human benefits for our well-being in an urban context. And this research has been taking so much of my energy and over the last decade that we ended up having to form a spin-off company, an environmental consultancy spin-off company that applied the science that I'm about to show you so that we can quickly meet the demands of planners, urban planners, land planners, and decision makers in the ministries to this challenge. So many of you will be familiar with what ecological connectivity is. We're trying to remove the limits to movement imposed by fragmentation of landscapes and by infrastructure that we lay down in our landscapes like roads that tend to inhibit movement. But it's also not just about the movement of animals and plants, but it's also about the flow of resources and the processes that interconnect these ecosystems. So we're trying to develop a view of the landscape that's fully connected in both senses. Recent research is showing us that when we start to put collars, we can track mammals. Here, this data is from mammals. This is a paper from Science that I was not involved in, but I find excellent and inspiring. We're showing how the 10-day long distance displacement of mammals is mediated by the human footprint index, right? As we go from a pristine environment on the left here up to urbanized land cover, you see we got 2.5 orders of magnitude change or decline in the movement of these 60-odd mammal species that this data set was based on. So we're now having direct evidence that human footprint has at different scales on movement. And what we know why we need the science, and in particular certain type of network science to manage connectivity, is that not all loss of connectivity is equal. So what you're seeing here is 3 identical made up landscapes, but I'm changing the rule by which patches are deleted from the landscape. All right, on the left, we're using a rule that's based on removing patches of high centrality, patches that are often in the middle of a series of movements that a species makes through the network. And on the left, I'm deleting species preferentially that have high betweenness centrality. Here we're losing patches in preference of their low betweenness centrality. And on the right, we're losing species, habitat patches, we're losing them randomly. And you can see that the moment at which we get the complete fragmentation of the landscape is very different in the three cases. And in general, we don't actually know how humans, from this point of view, how humans are fragmenting the networks of habitat that we're trying to protect. And in the reverse case, we want to be able to inform how to reconnect fragmented landscapes using these kinds of indicators and measures. So connectivity conservation has now become a major part of the IUCN as a connectivity conservation specialist group. And something that's become very clear as we try to apply this science is that we need to be able to provide relevant information that supports action of individuals, communities, institutions, and businesses as they turn their gaze to the ecological connectivity challenge. So the approach that we take is this network approach that I've alluded to in a couple of slides ago, where the science is that we identify a target territory or location. It could be anything from a local town or municipality all the way up to the eastern seaboard of the US and Canada. And what the algorithms that we apply and the models that we apply, what they do is that they identify viable networks of habitat. By viable, I mean as defined by the connectivity or the movement of individuals among patches, within patches, among patches, and also from the point of view of the traversability. If we're thinking about long-term connectivity, as species try to traverse landscapes under climate change, we have to try to manage the longer-term connectivity as well. And then our algorithms then allow you we apply sort of multi-criteria prioritization that allows you to prioritize the patches we need to protect first, right? Or the order in which we need to protect patches to create a connected network. So our workflow has different steps to it. There's a selecting species step, which is based on the ecology of the movement and the longevity or body size or various traits that predict movement capacity of species. And then we work through this flow here where we estimate habitat quality. There's a bit of French sneaking in here because our partners are Francophone, of course. And then once we know movement and habitat quality and it's distributed across the landscape, we can then quantify connectivity locally and regionally. And when we've done that, we can identify priorities of which elements of the network we need to protect, the corridors and the hubs or the nodes of the network. And then, of course, what's crucial is that we have to do it in a way that plans for future change. We have to roll out scenarios of how the landscape might change in the coming century using land use change modeling and regional climate models. We can update our advice. Based on the scenario that we derive from working in workshop with our stakeholders and partners, we can say, okay, under these scenarios, this is how we think you should distribute habitat and corridors to maintain connectivity out to 2100, for example. So I'll just show you some images of how our algorithm, what they do, what we generate from this science. So here in black, you're seeing the ecoregion of the St. Lawrence lowlands. This is Montreal down here in the left. This is Quebec City here. What you're seeing is that for these 5 target species, from shrews to martin, salamander, wood frog, and black bear, you're seeing how they perceive the connectivity of the landscape. And so in green are the habitat patches that they find of high quality, and in red, you're seeing the least cost paths that these individuals will take through the landscape. And so clearly, if we're going to apply an integrative biodiversity approach to connectivity, we need to take into account the functional perception, the functional movement and connectivity of these landscapes from the point of view of a range of species, right? So then we're building robust networks that are good for a range of life histories and uses of the landscape. So we generate these kinds of maps for each species to show you areas of high flux. So here we're using things like CircuitScape that some of you may know that are kind of flux measures of the probability of an individual moving through the landscape. And we can identify areas of high flux and areas of low flux. You'll not be surprised that the cities are areas of low flux because there's a high resistance in the landscape to movement and there's urban sprawl occurring. But the cool part we're able to do now, it's taken us a while to get here, is that now we can project land cover change using probabilistic land cover models from present day out, say 2010 out to 2100, and do this iteratively, sort of in a Monte Carlo way, where we can calculate the probability of every pixel, and there are hundreds of thousands, if not millions of pixels, depending on the resolution we use, how that's going to change, and how the land use change, therefore, would affect or impinge on connectivity. So we can use these scenarios then to show we can project fun dynamics like this. We can show how for a particular species the habitat patches are changing, the resistance of the landscape is changing, the centrality that I showed you before is changing, habitat suitability and current density. So all of these variables, these 5 variables I'm showing you here, go into our prioritization software, right? And then we can identify the key nodes, the key corridors to protect based on this dynamic representation of the future. So for example, here for the shrew, for the northern short-tailed shrew, we're showing how connectivity is changing every 20 odd years from 2010 out to 2100. Here is a little GIF of that, where you're seeing how connectivity is changing over time from the point of view of this species. And this is a powerful tool to communicate to a decision maker or somebody in a ministry or in a municipality or an NGO. We can use these kinds of tools to convey the change. And then we can even do more interesting tools where we can think of something like a Google Earth or Google Map, where we can now project the probability of movement for our different species here, and we turn on the fluxes that are, or the probabilities of certain types of movements will happen for different species. And then we can use this tool to inform a round table or a workshop where we're talking to different stakeholders at different spatial scales. So coming back to my obsession with scale and thinking about how scale should inform biodiversity science, well, here we're thinking about how scale should inform decisions, right? I can zoom into your backyard, I can zoom into my backyard and look to see how the connectivity is changing from the perspective of these different species. And we can add species as we want based on the availability of data and information. And then we can do these big summary reports. This is the most recent report where we're showing how these 5 different species here, how their percent change in mean connectivity is unfolding over the next century. Right, and you see different lines here that corresponds to different eco or physiographic regions of the St. Lawrence lowlands. In some regions, most regions, we're seeing a decline in percent connectivity, but in other areas, we're seeing an increase, right? Some of that's to do with the climate warming. Some species are going to do better under a warmer climate in the south of Quebec. And so we know that the realized connectivity for those species, the resistance of that landscape will go down, and so they'll tend to do better. So if we don't take a scenario approach, if we don't take into account the future, we're going to misestimate how connectivity is changing over space and time, and from the perspective of different species. And what we're able to do is then start to map this knowledge to targets, right? So we know in the last decade, the CBD had a 17% protected area target. So at the time when we did this research back, and we published this in Conservation Biology a few years back now, we showed how much connectivity could you maintain if you protected 17% of the remnant forest. All right, this was just forests and wetlands associated with forests. And this was just for the region around Montreal. You can see that in red, you're seeing the priorities to protect and how the prioritization differs from the case on the left here, when we only prioritize high habitat quality, versus the case when we prioritize habitat quality and connectivity. Now you can see that prioritization has this sort of contiguous structure to it, where we're building the network. And what we found, we were super happy about this finding, is that if we're smart about protecting connected networks, if we protect 17% of the top most connected locations and habitats, we can maintain 72% of the region's connectivity. So that's a huge return on an investment. Of course, that is not 100%. And so if we want to go beyond 72%, we need to go beyond the targets, at least the 17% target in this area. And of course, we can rescale the analysis and do it for the entire lowlands or the entirety of Quebec or the entirety of Canada. And we'll come to this sort of cost benefit analysis, or this perspective on the value of connectivity. And so I'm going to wrap up here by showing you a map that we just delivered, where we are now identifying the corridors and areas to prioritize here in bright green here, and the protected areas that are in yellow, and how we can vision connectivity for this region. Here you're seeing the protected areas adjacent to, it's north and south of the St. Lawrence lowlands, and how we propose connectivity should be planned for to allow not only connectivity within the lowlands, but to allow traversability of the lowlands under climate change over the next century. And if there's one thing that I've learned from this research of the application of connectivity science to policy, it's that we need strong partnerships, polycentric governance, where we're dealing with the issue at multiple spatial scales at different jurisdictions. And we have to do that by sustaining long-term relationships at the interface between science and application, between science and policy, between science and NGOs, between science and citizen groups. It's these partnerships that allow the cumulative gain in conservation objectives. And for something like connectivity, which is so integral to a landscape view, we really need lasting relationships if we're going to make a difference over time. And this is where the spin-off company that I set up, co-founded recently, has worked closely with the Quebec Centre for Biodiversity Science and then with the governments. We've been able to change the pace of advice, and that has allowed us to make inroads in terms of planning for conservation. So I'm going to stop here. I'm going to because I've taken too much time already. And I want to just leave you with a closing message that if you're interested in asking me about the arrival of GeoBon, it's come to Montreal this January. I'm now the co-chair of GeoBon. And so if you're interested in the problem of monitoring for connectivity, monitoring for biodiversity and ecosystem functioning, monitoring for species ranges, reach out to me. We're building the secretariat team right now at McGill. And so we're going to be trying to boost the application of GeoBon to Canada's context, to our provinces, to our federal network of reserves, thinking about how we can reboot Canada's monitoring and then place that in a global context when we think about how we can inch our way towards a global biodiversity monitoring system. So with that, thank you very much for your attention.